All right. Good evening, everyone. So today we are going to finish up Unit 1 by finishing up cell biology, getting all ready for that first unit test. I did send you away with homework last week, so hopefully you brought your strip sort activity for the endomembrane part of today, and I also asked you to draw some mitochondria and chloroplasts. So you're going to want to gather that stuff up so you can follow along today. Our focus is going to be eukaryotic cell biology, really looking at a review of some things you already know, and then sort of comparing what we know about eukaryotes to what we talked about with prokaryotes last time. This picture, probably pretty familiar, right? You've seen this type of picture lots of times. Most of you could probably identify most of the structures here. I'm going to show you a picture. I'm going to cold call people to give me their best guess as to what's being pointed at. And just to remind you that if I call your name, you can either, say what you think the structure is that's being pointed out, or you can pass, and that's also just fine. Okay, so we're just trying to go quickly around this little map of the cell. So let us begin with A1. We're going to go to Tiffany. That's this bit right here. A1. What is being pointed out in A1? Tiffany, if you're able to unmute and let us know what you think.
Nucleus?
Yes, the whole structure is the nucleus. And do you know what particular part is being called out here, this bit?
I don't know, like maybe the DNA?
That's a good guess. That's not the right answer, but it is an excellent guess. All right, let's just ask someone else to add on to a part of the nucleus, as Tiffany mentioned. Sean, what do you think is being called out here?
Is it the nuclear membrane?
It is the nuclear membrane. Good job. All right. What is Olusola? What is being pointed out in A2? And you can also see one of them here and here. What are these? Are you able to unmute Olusola? All right, it might be a microphone issue. I'm just going to go on to Michaela. What do you think is being called out in A2?
Is it the pore?
It is the nuclear pore. Good job. All right, A3, Marielle, what is this stuff here?
That's the chromatin.
That is the chromatin, the uncoiled DNA. And A4, Lucia, what would you say? A4, this dark spot.
Is that a nucleus?
That is the nucleolus. Good. All right. And as Tiffany mentioned, this whole entire structure is the nucleus. Okay. Let's next go to Lauren. B. What is B pointing to? It's pointing to several things that don't all look identical, but sort of have some things in common. What do you think, Lauren? What is B?
Is it the cytoskeleton?
Yes, that's an excellent answer. All right, we'll go to Kim for C. Now, C is sort of encompassing this whole area, the interior of the cell. What is C?
Is that the rough endoplasmic reticulum?
This part is, but this is kind of denoting the whole thing. What would you say then?
Is that the nuclear envelope, that part then?
This part is, yep.
Okay.
Okay, so we got rough ER, we got nuclear envelope. What about D? D, Katie. What would you say is D, this orange pancake thing?
The Golgi apparatus.
That is the Golgi. Good. E. It says there's a little pouch here. It says E. Assume that it is acidic. Catherine, what do you say?
Is that a lysosome?
It is a lysosome. Joshua, there's a process shown in F. What is that process?
I believe that's exocytosis.
It is exocytosis. Hannah, what's this little particle shown by the G, the line labeled G?
Are they ribosomes?
That is the ribosome. All right, Linda, what is this H? H is showing us what? What is this thing that's all around? Are you able to unmute? Okay. Oh, I see that Olusala did respond in the chat earlier. Okay. Fatu, I'm going to switch to you. What is H?
Hold on. H. I don't know why my thing is not showing everything.
That is all good. If you have to pass, that's completely fine if you can't see it.
I can see.
Yeah. Do you have a guess? All right, let's give our good friend Fatu a break here and go to Elizabeth. What's H, do you think?
Is it the outer membrane?
It is the membrane, the cell membrane. Good job. Danielle, what's I?
Mitochondria.
It is a mitochondrion. Good job. All right, for J, what's shown in J? For that, we're going to go to Casey.
You know, I was gonna say cilia, but I think that is wrong. So I'm gonna have to pass. I'm sorry.
All right, and passing is always permitted, so you don't have to feel bad about that. Thank you. Bisola, do you want to take a stab at what is shown in J, these pink things? Okay, there might be a microphone issue there. Let's go to Alexi. What is J, do you think?
Is that smooth ER?
It is the smooth ER. All right, and Bisola did answer in the chat, thank you. All right, what about K? K, Alexi, any thoughts about what K is?
Some kind of vesicle.
Some kind of vesicle is right. All right. Now, earlier we had talked about how this, as shown in L, is this rough ER. We can see with all the ribosomes attached. What about M? Ooh, let's go to Sean. What's M?
Is that the flagella?
It is the flagellum. All right, good job, guys. Okay, now we're in a different cell type. What cell type are we in? Put it into the chat. Good job. Yes, you guys, it is a plant. Raise your hand if you can say why you knew it was a plant. I suppose could also be an algae cell. How do we know? Raise your hand. Go ahead, Sean.
Is it because it has no cell wall?
It has a cell wall. Okay, so that's one clue. Excellent. Danielle, what would you add to that? What else tells you this could be a plant, could be an algae?
I was actually also going to say cell wall.
Okay, good. Lucia, what would you like to add?
It has a chloroplast.
Has these green chloroplasts for photosynthesis. Excellent. Anything else? What's shown in 2? What's this structure in 2? Put that into the chat. Oh, Joshua. Please be careful. Don't die for us. Yes, that is the vacuole. Giant vacuole is found in plants. Good. Okay. So those are animal and plants. Those are sort of the typical pictures you've seen of eukaryotic cells. Of course, when we talk about microbes that are eukaryotic, we're usually talking about single-celled or maybe a multi-celled algae or a small fungus. So, you know, it's going to look similar, but not necessarily identical. The answer key to all of those pictures is on Canvas. You can look at it if you are wanting to review. But just to be very clear, I'm not going to put this picture or that picture on a test and ask you to label it. I'm going to expect you already know this stuff, and that's sort of the basic point from which we're beginning. And I'm going to ask more interesting questions than what is this line pointing to? Okay, now this, again, you don't have to memorize what I'm about to show you here either, but this shows us what an electron microscope reveals about either animal cells, in this case, this is a bee cell in a human, or plant cells. And so we see all these beautiful structures. I would imagine that you'd have a decent shot at guessing what a lot of these things are, but I want you to put yourself in the shoes of brilliant scientists who are looking at these pictures for the first time, and they were like, wow, there's a lot of stuff in cells. We don't know what any of it is. So it is pretty amazing how far we've come in our understanding of cells, and a lot of that is thanks to electron microscopy. Okay, so let's talk about the eukaryotic cell wall. Now, in prokaryotes, the cell wall had a particular function. Is that the same function as eukaryotic cells have? And if so, what is that function? Okay, so some of the function provided by the cell wall in all cell types that have one is providing a shape, right? So that's true. I would say that's a secondary function, although it is an important one. What else? Yes, protection. Protection from what? Especially in microbes, what is that cell wall protecting the cell from? From being in a hypotonic environment, which could potentially lead to water rushing in and causing what we call osmotic lysis, basically bursting because you have so much water rushing into you. Okay, so protection is the primary goal of the cell wall in microbial eukaryotes as it is in microbial prokaryotes. Which eukaryotes have a cell wall? Write that into the chat. Right, plants, yep. Algae, fungi, not animal cells. Animal cells have no cell wall. And what is it though? Right, so plants, algae, fungi, and these are the ones that would be microscopic, not animals, not protozoans, because remember, these are single-celled eukaryotes that are animal-like in that they don't have a cell wall and they typically are motile. The composition of the cell wall, we remember what it was in prokaryotes, right? In bacteria in particular, what was the major chemical component of the cell wall in bacteria? In bacteria, what was that major cell component? Yes, that was peptidoglycan. And now in the eukaryotic world, is it the same? It is not. What is it? What is the cell wall made of? If it's not peptidoglycan in a eukaryotic cell, what is it? The cell wall. I know, Joshua, please don't feel that you have to text and drive. I will gather your thoughts with my mental powers. Okay, yes, polymers of glucose. Eukaryotic cell walls primarily composed of glucose polymers. And so in plants and algae, which are plant-like microbes, the cell wall is composed of cellulose, but it's a polymer of glucose. And in fungi, including yeast, the cell wall is made of chitin, which is also primarily composed of glucose. Of course, we talked about penicillin. It's a miracle drug. It doesn't harm human cells. Why not? Because what is it targeting? Okay, Danielle, good luck. What is penicillin targeting specifically? Yeah, that's right. Specifically, it targets the cross-links in peptidoglycan of gram-positives. Human cells don't have that. They don't have a cell wall. They don't have peptidoglycan. So let me ask you this question. Could you use penicillin as a treatment for a fungal infection? Yes or no? And if you're not sure, that's absolutely fine. You're always welcome to say that. All right, so majority of people are saying no, that's accurate. So tell me why. Raise your hand if you think you can explain why penicillin would not be effective against a fungal infection. Sean, go ahead.
I don't know if it's true, but I heard a long time ago that penicillin was developed from like mold, which could be the problem why it wouldn't work on fungi.
Yeah, so that's certainly kind of getting at the answer, right? So it's actually a mold that produces penicillin. So you would certainly imagine that it's not going to produce something that would hurt itself. We can be more specific. So that's a great start to that answer. Alexi, what would you like to add?
There's no peptidoglycan cross bridges in the cell wall of fungi.
Yeah, fungi have a cell wall, true, but they don't have peptidoglycan, and that's the target of penicillin. So they don't have the thing that penicillin attacks. So penicillin's not going to do anything to them. And so it makes sense, as Sean pointed out, that actually a mold is what produces the penicillin. Penicillin was used by mold to kill bacteria, their competitors, right? And so they're not going to make a drug that would kill them. But the molecular reason is penicillin attacks a particular molecule that's only found in bacteria. Okay. Now, other things that you can see on a eukaryotic cell that may appear to be familiar, right? There are flagella on prokaryotic cells, and there are also flagella on eukaryotic cells. And then there's also these cilia. And these cilia kind of look a little bit like pili and fimbriae. Looking at these pictures, it can be sort of tempting to kind of say, oh, that's like a prokaryotic flagellum. Oh, that's like prokaryotic pili. But actually, you have to remember this very important thing, which is that this cell is much bigger than a bacterium. Consequently, the flagella, which is to scale to the cell, a eukaryotic flagellum is going to be much, much bigger than a prokaryotic one. So you might expect that there's going to be some differences. Now, in terms of the function, right? The function of a prokaryotic flagellum is motility. What's the function of a eukaryotic flagellum? Okay, Marielle, good luck. Yeah, the function of a eukaryotic flagellum is just the same as a prokaryotic one, to help the cell move around, and especially true in microbes, right? There's some eukaryotic flagella and cilia in macroscopic multi-celled eukaryotes. It's more complicated, but in the microbial world, this is simple enough. Now, the structure, what is a eukaryotic flagellum made of? The cilia is actually made of the exact same thing. What is its structure? Eukaryotic flagella and cilia, how would you describe their structure? What are they made of? And if you don't know, putting a question mark is an excellent way to answer the question. As much as you can remember is an excellent contribution. Thanks for all of your answers and engagement. Just stay with me and stay awake. That's the name of the game, right? Okay, good, good. Keep those answers coming. Excellent, excellent. Okay. So as some of you have correctly pointed out, It's something called a 9+2 array of microtubules. What does that mean? Well, microtubules are hollow tubes. They're one of the cytoskeletal elements in eukaryotic cells. They're long tubes, but there's not just one. There are 9 pairs in a circle with 2 in the middle. And you might be like, why is that structure there? Why does it have to be so complicated? There are motor proteins attaching these to each other and that move. And this is actually what causes the motility of the flagellum. So without getting into much more detail, let's just say this. Remember that a microtubule is about 25 nanometers wide, which suggests that this whole structure is something on the order of 200 or 300 nanometers wide. Certainly big enough, like close to, but just a little bit bigger than the limit of resolution. So we should be able to, under the right circumstances, see this on a light microscope. We can't see a prokaryotic flagellum, at least not normally, because how big is it? How big is that prokaryotic flagellum? That's right. Nanometers, Elizabeth. Your number is right, but your units are wrong. Good answer. Just fix those units. Yeah. All right, guys. So remember, it's 15 nanometers. So it's more than 10 times smaller or skinnier, right? Now, the other difference is in the way that it moves, right? What was the movement of a prokaryotic flagellum? Last week, we talked about how prokaryotic flagella move. How do we describe what prokaryotic flagella do? Prokaryotic flagella, like on bacteria, they rotate, right? That's right. Like a propeller on a boat, Joshua, exactly. Now, what about these eukaryotic flagella? Think about that iconic picture of a sperm, swimming through liquid. It's wave-like or whip-like, right? Yeah, exactly. So prokaryotic flagella and eukaryotic flagella share function. They're both for motility, but they're structurally different. For example, just at the simplest level, the prokaryotic flagellum is much, much smaller. They're made of different protein, right? Microtubules in eukaryotes, flagellin in prokaryotes, and their way they move is different. Okay, prokaryotes had a glycocalyx. Remember that very external optional layer made of sugar? And there were 2 types in prokaryotes. Remember the capsule and the slime layer? What about eukaryotes? Do eukaryotic cells have a glycocalyx? Yeah, they don't always, but they can and often do. And just why do we care about it in microbiology in particular, aside from the fact that some eukaryotic microbes have one? Actually, the more important thing is in multi-celled eukaryotes like ourselves, that glycocalyx is sort of a part of the mucous membrane, is often the first line of contact with pathogens. Okay, let's talk about the cell membrane. Now, there are some things that the eukaryotic cell membrane shares with the prokaryotic cell membranes, actually a lot of things. For example, its function. The function is the same. What is that function? All right, nice job, you guys. So all cell membranes have the following function in common. Their important function is exchange, or a better way to say that, some of you said, is a selective permeability. So exchange and communication, allowing nutrients in, letting waste out, and finding out about the world, and also do provide a barrier function to separate the outside from the inside. That's a shared function. Structurally, they're similar. They're phospholipid bilayer and embedded membrane proteins. That's also the same. But there are some important differences, okay? So one of the things that we talked about last time is that bacteria and other prokaryotes often put electron transport chains and ATP synthase in their cell membrane in order to facilitate respiration and photosynthesis and other major biochemical pathways. But that's not found in eukaryotes because they delegate that to mitochondria and chloroplasts. But in the eukaryotic cell membrane, there's a function that's not present in prokaryotes called endocytosis. This is really important because remember, the eukaryotic cell is much, much bigger than a prokaryotic cell. One of the challenges as cells get bigger is that they need to exchange much more efficiently with the world because the interior contents are getting so massive and you need to provide the right materials to support that. Endocytosis allows that to happen because we can bring in massive amounts of material all at once, a whole protein or whole complex of proteins. In the case of phagocytes, your white blood cells that protect you from bacteria, entire cells can get engulfed by a phagocyte. So endocytosis is an amazing thing. Prokaryotes can't do that. If they want to bring something in from the outside, this is how they can do it. They have one protein or maybe a bunch of proteins that form a channel or a pore or a transporter, and they can bring in one ion, one sugar, one amino acid, okay? So it's very different. Now, there are also some structural differences. Yes, both types of cell membranes have a phospholipid bilayer and membrane proteins, but eukaryotic cell membranes additionally have steroids. In animal cells, it's cholesterol. In fungal cells, it's ergosterol, and plants have phytosterols. All right, now we come to the cytoskeleton. Do prokaryotic cells have a cytoskeleton? That's right, they do not. That is a eukaryotic innovation. Now, what is, and I should say, what are the functions of the cytoskeleton? Anything that you remember that they do? The cytoskeletal filaments, in which there are 3 types, actually. Structural support, shape of the cell. Anything else? Yeah, they facilitate cell division. They keep the contents organized, which is important because we get a lot of stuff inside of the eukaryotic cell, lots of organelles, lots of moving parts. Movement of the entire cell and movement of things within the cell. Good. So movement of the whole cell, movement within the cell, shape of the cell, the organization of the contents of the cell, and cell division are all facilitated by the cytoskeleton. What's it made of? Correct. It's made of protein. Now, more specifically, there's 3 types of cytoskeleton. Microtubules are made of a protein called tubulin. Microfilaments are made of a protein called actin. And then the intermediate filaments are made of a variety of different proteins depending on the cell type. For example, skin cells have keratin. So here's like a little chart that kind of summarizes all that stuff, like their size, their organization, their overall way that the subunits are organized, what they do, that kind of stuff. So I'm going to show you, so there's the 3 cytoskeletal filament types. Here is a description of their functions and where they're found, and then you're going to guess what I'm showing you. So if we were to fluorescently label just this one filament, we would see it organized often in a kind of perpendicular crisscross pattern at the edges of the cell where it facilitates crawling movements. And it looks like 2 strands of wire twisted together. What is this and how big is it? Give you a second to all weigh in on what you think is being depicted here. So this is the microfilaments, also known as actin, and it is 7 to 8 nanometers wide. Okay, so that's the smallest one. All right, now over here, we've got a type of cytoskeletal network that has organizing centers from which the filament grows. It grows out like hair from someone's head. It grows out. Usually the organizing center is near the nucleus in one spot, and so it grows out from there in all directions. This is what, and how big is it? Yep. That's right. These are microtubules, hollow tubes made of tubulin. These little round subunits are tubulin protein. And they're about 25 nanometers wide, the biggest ones. The last type is organized kind of like a rope-like network, kind of perinuclear all the way around, but not from an organizing center. And when you look at it, its organization is more fibrous. It provides kind of strength. Cells and tissues are connected using this. What is it? Yeah, that's right. These are the so-called intermediate filaments because they're intermediate in size and they're about 10 nanometers. Good. The one thing that I just want to kind of point out to sort of add to, like, this is stuff that's probably hopefully review for you, but look at these three patterns, where these filaments are located. It kind of helps to illustrate that, yeah, the whole cell is full of these networks, right? But they are actually different from each other, not just in how big they are, what they're made of, or what their purpose is, but actually where they're located. And we can really see this if we use an a fluorescent microscope and we label the proteins with different colors. We can really see this nicely under a microscope. Next, we come to the ribosomes, the prokaryotic ribosome. Its function was protein synthesis. Its structure was the 70S size, actually means density. And it's made of two subunits, the large 50S and the small 30S subunit. Is the function of the eukaryotic ribosome the same? Yes or no? Oh, that's funny. Well, we saw that the intermediate filament looks like a true. I think so too. All right, yes, the function is the same. It's also for protein synthesis. Is it structurally the same? And if not, what is it? How would you describe it? If it's not 70S with a 50 and 30, what is it? Yes, that's right. Correct. It is 80S with a 60 and 40 subunit. Also, the location, a little bit different. Prokaryotes have a cytoplasm. That's where you'll find their ribosomes. Eukaryotes have a cytoplasm, and they also have, and that's where a lot of the ribosomes are, but you'll also find ribosomes attached to the endoplasmic reticulum. Question, are there 70S ribosomes in eukaryotes? Yes or no? Yes, there are. Good job, you guys. Okay, there are 70S ribosomes in eukaryotes. And so what is going on? Because eukaryotic ribosomes are 80S. Where are these guys? Raise your hand. Where are they? What are they doing inside of a eukaryotic cell? Raise your hand if you want to answer that. Catherine, go ahead.
Are they in the chloroplasts in mitochondria?
That is correct.
I wasn't sure.
Yes. So, you know, it would also be fair to say they're not found in every single eukaryotic cell, right? Because not every eukaryote has a chloroplast, and there are even some eukaryotes don't have mitochondria. You might not know that because we have mitochondria and our cells need it, but there aren't any mitochondria in some species of yeast, for example. But those eukaryotic cells that do have either a chloroplast or mitochondria, or in some cases both, their chloroplasts and mitochondria have 70S ribosomes in them. And that becomes important later on when we talk about the endosymbiotic theory, which explains the origin of these organelles. So there are 70S ribosomes in chloroplasts and mitochondria. So eukaryotes that have these organelles will also have 70S. So these cells are going to have 80S in the cytoplasm, 80S attached to the ER, and 70S in the chloroplast and or mitochondria. Okay, we're not taking a break yet. We are going to just have you quickly name it a eukaryotic organelle. Just put that into the chat, any eukaryotic organelle that you can think of, and I will comment on them as I see them. Endoplasmic reticulum, yes. Nucleus, yes. Golgi complex, yes. Lysosome, yes. Ribosomes are not actually technically organelles. They are not bounded by membrane. And one way to remember that is that all cells have ribosomes. So they are not organelles, which only are found in eukaryotes. Lysosome, yes. Mitochondria, centrosome, no. Although it is eukaryotic, it's not bounded by a membrane technically. Peroxisome, vacuole, yes. And the plasma membrane is not considered an organelle. It is the main membrane. All right. Now, I'm not going to list everything, but I am going to show you the list of the things we're going to care about today. So in purple are the ones that are considered part of, or at least important for, the endomembrane system. And then down here are the energy generating ones. Okay, we're going to talk about those today. Let's start with the endomembrane system. So sort of traditionally, a purist would say the endomembrane system consists of the rough ER, smooth ER, vesicles that transport between these two, the Golgi and lysosomes. Some people additionally include the nucleus because actually technically it's connected to the rough ER and is important for the first step of what the endomembrane system does. So let's just briefly remind ourselves what these organelles do individually, and then we're going to talk about how they work together as a system. So the nucleus, of course, is where the DNA is found, the instructions for proteins. And this is where, when you want to make a particular protein, you transcribe the DNA to a piece of messenger RNA. The rough ER is where protein is made, at least where some cellular protein is made. That's a hint. The smooth ER is where lipids are made. And then there are vesicles that move stuff between the ER and the Golgi, between the Golgi and the membrane. Things that end up in the Golgi are modified. That's the purpose of going to the Golgi, is to get modified and sorted and packaged. Remember that? But the modification is done to stuff that's going to leave the cell or going to be on the surface of the cell. If you're going to leave the cell or be on the surface, you need to be decorated with sugar, a process known as glycosylation. So there are enzymes in the Golgi that will do that. And you may not know this, but I'm telling you that lysosomes are also made from the Golgi. So the Golgi will make vesicles that send stuff to the membrane, but it will also make vesicles that become lysosomes. So let's now talk about how these organelles work together, because they're not really operating alone. They have, I mean, really everything in your cell is kind of operating as a great big system, but the endomembrane system in particular is how the ER, Golgi, and vesicles all kind of work together to accomplish some common goals that they couldn't do on their own. So what are those goals? Let's start with this. If you're a eukaryotic cell and you want to export some protein so it goes outside and somebody else gets to use it, you have to use the endomembrane system to make exported protein. So as an example, you guys have been studying human cell biology primarily. From A and P, can you think of any tissues that would, of course, contain cells that make protein the rest of the body uses? Think about anything like protein hormones, defensive proteins, anything that a cell in the human body makes. It makes a protein for export. Okay, the pancreatic cells. Yes, Alexi, what did they make? What protein did they export?
I think they do both insulin and ghrelin. Is that right?
Yeah, they do. So insulin is what I was thinking of, and there are other hormones as well that are produced by the pancreas, including glucagon. But yes, insulin made by pancreatic cells is an excellent example. Insulin is just this little shorty little protein. The pancreatic cells don't make that protein for themselves. They make it and they send it outside. What about defensive proteins that protect you from disease? What's the name of those guys? A defensive, yeah, antibodies. Who makes those? Who makes those antibodies? Lymphocytes, which one? Which specific lymphocyte? It's not the T cell, that's a good guess. We'll talk about T cells in a second. The B cell makes antibodies. Good. Again, the B cell makes antibodies, a protein that it sends outside. Okay, how does it make it? The endomembrane system. Okay, membrane proteins. Now, we've just talked about how the cell membrane always has proteins in it. There's lots of proteins. There's protein channels, protein transporters, there's receptors to receive information. All of those proteins get to the membrane by being made in the endomembrane system. Also, you have to continually make new membrane lipids. That comes from the smooth ER. Lysosomes. I just mentioned this, not everybody knows this, but lysosomes don't just magically appear out of nowhere, like boom, I'm a lysosome. They are made by having proteins made through the endomembrane system packaged into a vesicle that becomes a lysosome. And last but not least, in the endoplasmic reticulum and in the Golgi, there's biochemistry that has to happen. Those enzymes, those ER and Golgi resident enzymes, don't just magically appear in those compartments. They are made by the endomembrane system and sent to where they're supposed to go. All right, so let me ask you this question because we haven't talked about it yet. There are also proteins that the cell uses in the cytoplasm, okay? So for example, glycolysis is an 11-step biochemical process that requires a bunch of enzymes that are proteins that do their work in the cytoplasm. Did the endomembrane system make those enzymes? Yes or no? And it's fine to put a question mark if you don't know. I'm asking you this because, you know, when you learn about cell biology, one of the things you learn about is that the rough ER makes protein, but it's kind of overshadows the fact that that's not where all of the protein is made. So actually, the cell has lots of proteins that are made in the cytoplasm and that remain in the cytoplasm, never go through the endomembrane system. Stuff goes through the endomembrane system only if it has to, okay? So the answer is no, and let's just briefly talk about it. We'll revisit this in the genetics section, but there are proteins that are not made in the endomembrane system, are not made at the ER, at the rough ER. So how does that work? The DNA instructions for those proteins is in the nucleus. That's where you transcribe that into mRNA. The mRNA leaves through the nuclear pore. It's grabbed by a free, not attached ribosome, made into protein in the cytoplasm. Boom, you're done. No endomembrane system had to get involved. Now, let's talk about the proteins that do go through the endomembrane system. For example, exported proteins or membrane proteins. So starting with, like, say, an antibody being made by a B cell, what has to happen? The instructions for that antibody are in the nucleus. They get made into mRNA that leaves through the nuclear pore. And it then gets made in the rough ER. And then what happens is it's packaged into a vesicle, sent to the Golgi, glycosylated, packaged into another vesicle, sent to the membrane, and it leaves by the process of exocytosis. Much more complicated, right? Same way that membrane proteins arrive at the membrane. What happens is the instructions for the membrane protein are found in the nucleus. The DNA is transcribed to mRNA. It leaves the nuclear pore. It's grabbed by a ribosome and made in the membrane of the ER. Packaged into a vesicle. Whoops, I went backwards. Sorry, guys. Packaged into a vesicle. Because it's in the membrane of the ER, it's packaged into the membrane of the vesicle. That vesicle fuses with the Golgi. Now it's in the membrane of the Golgi. The Golgi is where the part that's sticking into the interior of the Golgi is glycosylated. packaged into another vesicle. Now it's in the membrane of the vesicle. It goes to the membrane. It's in the membrane of the vesicle. When the vesicle fuses in the process of exocytosis, the protein is delivered to the cell membrane. Okay, so just to recap, for exported proteins, mRNA is made into protein in the lumen of the ER, the interior of the ER, and then it's packaged up into a vesicle, sent to the Golgi, ends up in the interior of the Golgi where it's glycosylated, packaged up into another vesicle, sent to the cell membrane, and sent outside by the process of exocytosis. Let's briefly check in and see if we understood some of that. Which made it, the endomembrane system or a cytoplasmic ribosome? Antibodies made by B cells. Endomembrane system made it, or cytoplasmic ribosome? Correct. Endomembrane system had to step in. All right, T cells. T cells have receptors on their surface. A famous one on a T helper cell is called CD4, and that is a membrane protein. So do membrane proteins get made by the endomembrane system or by cytoplasmic ribosomes? Oh, I'm sorry about that, Marielle, that sucks. Correct, it's the endomembrane system again. Okay, you're inside the Golgi. You have to glycosylate a bunch of things that are going outside. The enzyme that's inside the Golgi, how did it get there? Was it made by the endomembrane system or by a cytoplasmic ribosome? Great. Okay, so most of you are guessing cytoplasmic ribosome, and you're wrong, but don't feel bad because that's what I would have guessed too. Why? Well, because we think, oh, cytoplasmic ribosomes are going to make stuff the cell keeps. Not exactly. Okay, cytoplasmic ribosomes make stuff the cell is going to use in the cytoplasm. But if it's something that has to be inside of the Golgi, send it through the endomembrane system. So the endomembrane system makes this too. Okay, what about a glycolysis enzyme that's going to be used in the cytoplasm? Yes, this time cytoplasm is the right answer. Fabulous. Okay. Before the break, we're going to tackle the homework that I set for you. So let me explain what this was. These are some things the endomembrane system can do. When it's working together, here are some things that can happen. Remember, the endomembrane system can accomplish 4 different things. It can make exported proteins, membrane proteins, resident ER and Golgi proteins, and lysosomes. For each of those goals, it does not need to use all of these steps. Your job for each of the prompts I'm about to give you is to decide which steps are required in what order to accomplish the goal, okay? So the first thing that I asked you to do for your homework was, which of these things in what order is required for B cells to make antibodies for export? Put your answer into the chat. And if you need a minute, I'll give you a minute. If you have no clue whatsoever, you can put a question mark, but give it a shot. You can also read the chat, see what other people are saying, and see if what they're saying makes sense to you. And you can steal their answer. So one time in this class, you're allowed to cheat. It's not cheating, it's just learning. Danielle, did you have a question?
All right, my computer is still bugging out really bad and it won't let me see the chat or go into the chat at all.
Okay.
During the break, I'm going to restart it, but I can't look at anything right now.
Okay, are you able to see my screen?
Yes.
Okay, sweet. Well, don't sweat about it too much. Just sort of play along in your mind. And at the break, which is coming very soon, hopefully you'll be able to leave and come back, and fingers crossed it will work.
I hope so. I'm really sorry. I haven't been able to like comment on anything.
That is completely fine. I do see you here, and so don't fret too much. It's okay. Thank you for mentioning that, though. I appreciate you. Okay, let's have a look at these answers, guys. Good job. The majority of people answered 1, 3, 4, 6. That is the correct answer. Let's see why. So I want to send this protein outside the cell. I don't want to put it in the membrane. I don't want to put it in the lysosome. I want to send it all the way outside. So the first thing I do is I take the instructions, make it an mRNA, and I translate that into antibody protein in the interior of the ER, what we call the ER lumen. Package it up in a vesicle and send it to the Golgi. Decorate it with sugar because it's got to go outside. Package it up in another vesicle, send it to the plasma membrane, and exocytosis dumps it outside. Good. Okay, so you're going to have the break to think about this one. And then when you come back, we'll talk about it. And then for the final prompts, we'll do a breakout. So I'm going to give you your break to think about how you would do this one. So what is it? A T helper cell has a protein called CD4 in the membrane. Think about how you would get it there using these, which of these steps in what order. Not all of them are required. So which steps in what order? Think about that. If you get an answer early, you can put it into the chat whenever you want, but we will come back in 10 minutes and recap. So actually 8 minutes. I'm going to see you at the top of the hour at 7 o'clock. Okay, so see you at 7 for the answer to this question. Welcome back. All right, I asked you to think about this on the break. Mariella, you're fine. I asked you to think about how you would get a protein to the membrane. So put your answers into the chat now. All right, good job, everyone. So I don't see the 100% right answer yet, but what I am seeing that makes me very happy is that most of you are starting with the correct step. You've noticed how you need to start this thing off in order to get to the right spot. So let's just wait for any more answers. If you're not sure, if you had no idea, you can put in a question mark, but just do engage with it just so I kind of know not to wait for you. I've got about a dozen answers. I've got 18 people, so a few people haven't weighed in yet. Okay, good. Great. Okay, so here is the answer. So as many of you suspected, a really key part to this is when you go to make the protein, because you want it to end up in the cell membrane, instead of putting it into the interior of the ER, you put it into the membrane of the ER as you're making it. Why that's important is that means that when you go to make the vesicle to send it to the Golgi, it's actually going to end up in the membrane of the vesicle. When that vesicle fuses with the Golgi, it's going to end up in the membrane of the Golgi. It's going to still be glycosylated because it is going outside the cell. But since it's in the membrane of the Golgi, when it's made up into a vesicle to be sent to the plasma membrane, it's now in the membrane of the vesicle. And when that vesicle fuses with the plasma membrane, instead of being dumped outside, which would happen if it was entirely in the interior of the ER, it fuses with the membrane and becomes part of the membrane. And that's how you end up with a membrane protein. So good job, everyone. Some of you didn't know that you had to glycosylate it. Well, that's true of anything that either is leaving the cell altogether or is going to be displayed on the surface. Okay, next prompt, and I'm going to send you to breakout rooms for this one. So you might want to make sure that you have this strip sort in hard copy or up on your computer, or that you take a screenshot of it really quick so that you have it. Okay, but the prompt is this. Okay, so now you'll notice that I'm using all these cells. It's not on accident. When we go to immunology at the end, we're going to talk about all these cells. So now we're going to think about a white blood cell called a phagocyte. Its job is to go around engulfing things, for example, bacteria, viruses, toxic proteins, etc. So when it engulfs them through the process of endocytosis, makes a vesicle, which then fuses with lysosomes to digest. So it has lots of lysosomes. So how do those lysosomes get made? Remember, the endomembrane system is required. What are the steps? Think about it. And I'm going to send you to breakout rooms to talk about it. Let me just create those breakout rooms, and you can talk to each other about what you think are the steps required to make a lysosome with the endomembrane system. Off you go. Now, before I send you away, once your group is finished, you have an answer that you all agree on, come back to the main room so I know you're done. All right, away you go. All right, our breakout rooms have now closed. Welcome back to the main session, guys. You have a second if you're just joining us to put your answer into the chat. How do you make a lysosome? What is your guess? Okay, so lysosomes contain a bunch of proteins, and they're an organelle that has a specialized function that remains inside of the cell. So it doesn't ever go to the plasma membrane, doesn't get dumped outside, doesn't join the plasma membrane. It therefore doesn't need to be glycosylated because it's not going outside. So we can eliminate that step. The enzymes, for the most part, are in the interior of the lysosome, so we don't need to insert anything into the membrane. So our first step is going to be make the proteins. And there's a lot of different lysosomal enzymes, but make those enzymes in the interior of the ER. Package them up, send them to the Golgi. Then what happens to them there is the Golgi says, oh, you're lysosomal enzymes. I'm going to put you all over here. I'm going to make a vesicle out of all of you. That vesicle becomes the lysosome. Okay, so sorting and packaging into a lysosome, and then you're done. Boom, you don't have to go to the plasma membrane. You don't need to be glycosylated. You're just done. I mean, there's a few other things that happen, but you know, for our purposes, we're done. Okay, last but not least, and let's just, instead of sending you away to breakouts, let's just think about this together. Remember I told you that there are enzymes inside, say the Golgi, right, that do this glycosylation process. How do you get that enzyme into the Golgi? Which steps do you use? You lose this Squid Game, Joshua. I'm glad it was fun speculating with Lusola. Yes, it should be fun, right? You don't have to be right all the time. You just have to try. How do you make a glycosylation enzyme that ends up in the Golgi? What steps are required, do you think? Just put your thoughts in there. And luckily this isn't a Squid Game really, because you know, if your numbers are wrong, nobody dies. Hopefully. Certainly not because of anything I would choose to do. Okay. Good, keep those answers coming. Great. Okay, so let's think about it for a second. For the all intents and purposes, at least for this conversation, we're going to assume that the enzyme is in the interior of the Golgi. It doesn't need to be in the membrane, it's just hanging out in the inside of the Golgi. If you know a lot about cell biology, you can have a conversation with me about alternatives, and you would not be wrong, but let's just assume it's going to be in the inside of the Golgi entirely. So we just make it like normal in the ER lumen. Then we package it up in a vesicle and send it to the Golgi, and we're done. It's not going outside, so it doesn't need to be glycosylated. It's not going to the lysosome. Doesn't have to get further transport. So it's just boom, right? You make it, you send it to the Golgi, and you're done. All right, so if not all of this is crystal clear, I don't want you to fret about that right this second. Okay, this activity is available to you. Go back and watch the video lectures on this topic and then reapproach this, see if it makes more sense. And then at any point you could send me an email asking for clarification about something, or you can just ask me in the Q&A on Monday. Like I still am kind of stuck on how do you make whatever. Just sit with it a little bit, it will make more sense. Also, I have time at the end of class today to go over it again with anybody who needs that is absolutely fine. So if you're feeling confident, great. If you're not feeling confident, that's okay too. I can stick around today or we can talk about it again on Monday. For right now, I want to move on to these two organelles, mitochondria and chloroplast. And I want to tell you the two reasons that we need to talk about these organelles in particular. First of all, they are the place where important metabolic processes happen in some eukaryotic cell types. And then we want to be able to compare that to what happens when these same processes occur in a prokaryotic cell, which does not have these organelles. So in eukaryotes that do cellular respiration, the process happens in the mitochondrion. And in eukaryotes that do photosynthesis, the process happens in the chloroplast. When we get to metabolism, we're going to talk about those processes again. And you know, for some people, metabolism is very challenging because it's a bunch of reactions and it can seem extremely abstract. I find it's much easier to remember and understand if you have a visual idea of where stuff is happening. Just as an example, okay, let's suppose you didn't know anything about human life and you're coming from another planet. I told you that people spend their lives eating, sleeping, and pooping, okay, and watching TV, maybe going to work. It might be a lot easier to understand and remember if you had a map of the house and you saw that there was a bedroom and that's where the people go to sleep. And you saw there was a kitchen and that's where they make their meals. And there's a bathroom and there's a couch and a living room, right? So it's not different when we're talking about these biochemical processes. They're not just happening in limbo, they're happening in a place in the cell, and it makes sense where they're happening. So I want us to remember the structure of the mitochondria and chloroplast in particular so that we can map those biochemical processes onto it. So mitochondria, as you will remember, this should be a familiar picture. They have two membranes, an outer membrane and an inner membrane, right? And because of this structure, they also have two fluid-filled compartments that are separate from each other. The very interior one here is the matrix, right? This is fluid called the matrix. And then there's a compartment that's filled with fluid that's in between these two membranes, and that's called the intermembrane space. Okay, so that's kind of the structure of a mitochondrion that you hopefully remember, or maybe at some point knew and just have to re-dig out of your brain. But now let's talk about what is happening in this mitochondrion. So when you draw your picture, you know, make sure that you draw an outer membrane and an inner membrane and label the matrix, which is the most interior space, and the intermembrane space, which is between the two membranes. What is the pH of the matrix? If you're not sure, you can put a question mark, or you can guess, or you can Google it, but what do you think it is? And Sean, it looks like you have a question.
No, I hit the wrong button. I was going to say the pH was 8.
Awesome. Okay. Yes, everyone's putting 7 or 8, and that's accurate. Okay. Now it's actually closer to 8, but if you were to choose 7 or say neutral, I would accept that because the matrix is neutral-ish. It is a site of a lot of biochemistry, for example, the Krebs cycle. Okay, now the space in between the two membranes is called the intermembrane space. And what pH is that space usually? That's right. It's usually acidic between 2 and 4, okay? Acidic meaning what? What is accumulating in that intermembrane space to make it acidic? hydrogen ions. Right. Okay. Now, you'll remember also from metabolism that you studied before, respiration, which is what's happening in the mitochondria. Respiration, a big important part of it is the electron transport chain. Electrons are sort of loaded into the chain at the start. They flow through the chain. The energy from those electrons is harvested by the electron transport chain to pump protons. So let's start with, where is the electron transport chain located? And I'll just give you a hint up front that electron transport chains obviously handle electrons. In order for them to do their job and do it correctly, they have to be in a membrane. So which of these membranes is where you'll find the electron transport chain? Correct. The inner mitochondrial membrane. Let's look at that on this picture. Here it is. This inner folded up membrane is where the electron transport chain is located. Electrons flow through the chain. The energy that's released by that movement of electrons is used to pump protons across this membrane. So the act of transport using the energy from electrons of protons across this membrane is taking protons from where to where? Are the protons being pumped from the matrix into the intermembrane space, or are they being pumped from the intermembrane space into the matrix? Which direction are they actively being transported? Are they being pumped into the intermembrane space, or are they being pumped into the matrix? Which one is it? So far, we're kind of 50-50. So whichever way you go, you're going to be in good company with other people. You shouldn't be afraid to take your guess. It's more conceptual, Alexi. Yeah. The nitty-gritty details of each pathway, not so important. Yeah. Good. Okay. So I would say we're 50-50 kind of on the answer here. So this is exciting, right? The answer is, for the part, because the protons actually flow both ways. They go across and then they come back. But the part where they're being pumped actively using energy, sort of being pumped against their concentration gradient in order to make the compartment acidic, they're being pumped from the matrix into the intermembrane space, which is why the matrix is becoming slightly basic. It's being depleted of hydrogen ions, and the intermembrane space is becoming acidic. It's becoming full of hydrogen ions. Okay. So we've talked about the pH, we've talked about where this is, and we've talked about where protons go during respiration. Okay, so you want to remember that. That will help you understand how respiration works at the level that I want you to understand it when we get there. Let's talk about the chloroplast now. The chloroplast, like the mitochondrion, has two membranes, an outer and inner, but has a third set of membranes called the, what are these guys called? That's right, the thylakoid membranes, okay? They're little pouches. And so now we have really, the intermembrane space is not so important for chloroplasts, but we still do have two fluid-filled compartments that are important. There's the fluid that's kind of bathing thylakoids, that's on the outside of the thylakoids. What's that fluid called? That's right, that's the stroma. And then the inside of the thylakoid, you could call it the interior of the thylakoid, or you could call it the lumen of the thylakoid, right? So we've got these two fluid-filled compartments separated by the thylakoid membrane. All right. So you want to make sure that when you draw your chloroplast, you're kind of paying attention to, there's inner and outer membranes, there's thylakoid membranes, stroma, which is the fluid bathing the thylakoids, there's the interior of the thylakoid. What pH is the stroma? This is a compartment where lots of chemistry happens. It's kind of similar in concept to the matrix. A cycle happens here. It's not the Krebs cycle, but rather the Calvin cycle. And like the matrix, its pH is, yes, neutral-ish, pH 8. If you were to say 7 or neutral, I would still accept that, but it's actually closer to 8. Now, just like respiration, photosynthesis requires electron transport chains, and so that electrons can flow through and the energy from them can be converted into movement of protons. There's also photosystems that collect energy from light. The photosystems, they're like a solar panel. They absorb photons. The energy from the photons is used to energize electrons that were stolen from water molecules. Those energized electrons flow through the adjacent electron transport chain. Where are those machines that handle electrons and photons located? Again, they have to be in a membrane. We've got lots of membranes to choose from. Which one is it? I'll just wait for you to log in your answers. Good job, everybody. Okay. So a mix of answers, but if you answered thylakoid membrane, you are correct. That's where they are. Let's have a look at this diagram again. Dear, oh dear, there we go. So thylakoid membrane shown in green is where the photosystem is located and where the electron transport chains are located. Now, again, the photosystems are gathering energy to energize electrons, which are being fed to electron transport chains. Those electron transport chains in the thylakoid are actively pumping protons across this thylakoid membrane. On either side of the membrane, we've either got stroma or the lumen of the thylakoid. For this part where we're using energy to pump protons against their concentration gradient to acidify a compartment, where are those protons going to? Are they being pumped into the stroma or are they being pumped into the interior of the thylakoid? Great. So just like before, we've got two answers, like roughly, I would say even. People are like, maybe it's this, maybe it's that. Let me continue sort of giving you information so you can make a better and better educated guess. Don't forget that the stroma is usually neutral-ish. Biochemistry has to happen there. We don't want to acidify that compartment because then all the enzymes that facilitate the Calvin cycle are going to fall apart. Nothing's really going on in the interior of the thylakoid. So that would be a safe place to stow protons. So it is the protons are being pumped into to create a gradient in an acidic compartment inside the thylakoid, the interior or the lumen of the thylakoid. Okay. So we're going to talk about all of that again in metabolism. So just to remind you of what to sort of keep in mind, pay attention to. Where are the electron transport chains in mitochondria and chloroplasts? ATP synthase, by the way, is going to be found right next to the electron transport chains. Where in these organelles will you find the fluid-filled compartment that is a low pH? In the mitochondrion, it is the intermembrane space. In the chloroplast, it's the lumen or interior of the thylakoid. Where is the important biochemical cycle happening? The Krebs cycle in the mitochondrion is happening where? And the Krebs cycle in the mitochondrion is happening where? That's right. Krebs cycle in the mitochondrion, which is part of respiration, is happening in the yes, that's right. In everyone's favorite movie, The Matrix. Okay. And the Calvin cycle, it's an important part of photosynthesis, needs to happen in a chloroplast in a neutral-ish environment, is happening where? Yes, stroma, which does not make a snappy movie title, but I challenge anyone to make that screenplay and let's all watch it anyways, okay? Now, we all remember that prokaryotes don't have organelles. They don't have mitochondria. They don't have chloroplasts. In a eukaryotic cell, respiration is performed in the mitochondria and photosynthesis in the chloroplast. Can prokaryotes do either of these processes or can they not? Yes, Alexi. Awesome, Josh. I can't wait. You guys are getting me so excited. Can prokaryotes do these types of biochemical reactions or no? Excellent. I have indoctrinated you well. Yes, they can, even though they don't have the organelles that we commonly associate with these processes. Now, but there is a consequence for that, right? So where is the electron transport chain for either respiration or photosynthesis if there's no inner mitochondrial membrane and no thylakoid membrane? Where is the electron transport chain in the ATP synthase in a prokaryotic cell? They still need it. They still need it in a membrane. What is the membrane that they are you going to put it in? Right. You got it. It has to go in the only membrane available in a prokaryotic cell, the plasma membrane. Good job. Now, here's a question, okay? Think carefully about this. We have to pump protons across that membrane. That's how it works, right? Creating a proton gradient. So protons have to accumulate on one side. That is going to become an acidic, low pH environment. Which direction are we pumping our protons if we're a prokaryotic cell? Are we pumping them into the cytoplasm, or are we pumping them into the extracellular space? Right. The final frontier. These are the voyages of the, I don't know, someone who's less tired than me and more creative can finish that sentence. I love that show. I was raised on it. I'm like definitely a product of Star Trek. Okay, now I have the theme song playing in my head. Thank you for that. In the extracellular space, exactly. Why? Why is that important? What would happen if it was the other way around? You would acidify the cytoplasm where like glycolysis is happening and other things that we'll talk about in a second, destroying all those enzymes. So that's going to be a terrible idea. Okay, so it has to be that they're pumping protons outside the cell. In this hypothetical prokaryote that's doing respiration and photosynthesis, where are the enzymes for the Krebs cycle and the Calvin cycle located? There is no matrix, there is no stroma. Where are these cycles? It's not a membrane, though that's a great guess. Because remember, these cycles don't happen in a membrane, they happen in a fluid-filled compartment. What is the fluid-filled compartment that you have available in a prokaryotic cell? Yeah, the cytoplasm, exactly. Okay, now I'm going to move on to what seems like a different aspect of mitochondria and chloroplasts, but if you think about it, really isn't. This is a theory that explains the origin potentially of modern eukaryotic cells and the origin of the organelles, mitochondria, and chloroplasts. That theory is called the endosymbiotic theory. And who is the scientist who came up with it? This is a recent theory. The scientist who is credited with this theory actually was awarded the presidential medal by President Clinton in what I hope for most of you was your lifetime. If you're super, super young, maybe I'm wrong about that. Okay, so that dates me a little bit. Who came up with this theory? This is in your prep, so, and it's part of the history. You do have to know this. Thank you, Sean. You make me feel slightly less sad. Yes, that's right. It was Lynn Margulis. So you do have to know her. What does it propose? In essence, it proposes an explanation for the origin of mitochondria and chloroplasts and modern eukaryotic cells The idea is, how would you summarize it if you were to write it into the chat really quick? What is the sort of like basic thesis of this theory? Yes, Casey, you're right. Yes, yes, that's right, Casey. Yes, Alexi. Right. Okay. So I'm just going to read some of the statements because altogether they kind of make a good summary. The theory explains how mitochondria and chloroplast are derived from bacteria via endocytosis and internalization and then subsequent symbiosis. So the idea is that these organelles were once free-living bacteria that were engulfed by a larger cell. And these free-living bacteria were either able to do aerobic respiration or photosynthesis, and they provided those functions to the larger cell that they were engulfed by, and over evolutionary time became so interdependent that they lost their freedom of being independent, and the cell also became dependent on them and outsourced its own biochemistry to those organelles. So what is the evidence for this? Put any piece of evidence into the chat because there's several lines of evidence that support this. It's a very elegant theory. It's well supported by the evidence. Okay, so these organelles have their own DNA, which is a little bit unusual, and it's a circular piece of DNA, much like a bacterial DNA. The structure, size, and the way that these organelles reproduce, because they can, is very much like a prokaryote. They're small, and they reproduce by something that's kind of like binary fission. They're ribosomes, right? Their ribosomes are like prokaryotic ribosomes. They have their own ribosomes, first of all, and they're 7DS ribosomes. There are living intermediates. That's right. There are examples of intermediates that kind of show the transition step. The double membrane. There's two membranes. This is completely consistent with what would happen if a bacterium was engulfed by a cell, and instead of being destroyed, when you engulf something, you wrap another membrane around it. The way that it interacts with antibiotics affect these organelles in a similar way to the way they affect bacteria. Isn't it, Olusola? I love it so much. It is bonkers, Alexi. You're completely right. It's bonkers. Okay, so mitochondria and chloroplast both have DNA, and it's circular like a bacterium. They both have their own ribosomes, which are 7DS like prokaryotes. They have double membranes as if they were engulfed. They're similar in size, replication, and their effect, or the way they're affected by antibiotics, is similar. Okay. And so here's a picture of kind of the idea about how these occurred. These were free-living, respiring or photosynthetic bacteria engulfed by a larger cell, and over time became interdependent on that cell. Okay, so there's a couple of things I want to talk about before I do the final little activity here. I just want to make sure that I get this information out. So don't leave yet because we're not done, but I just want to make sure that I say something so that everybody has this info. Unit test 1 is next Wednesday. On Monday, we're going to do growth, the beginning of unit 2. So I'm going to tell you how to prepare for that. But I want to explain to you how the test works. So I'm going to open the Zoom early at 9:30. You don't have to be there at 9:30. You can come at 10 or 10:15 at the latest, but you want to bring your ID, picture ID. You're going to sign in like you always do, write your name into the chat. Okay. Turn your video on, which is unusual. Wait for me to call you. I'm going to call on people in the order I see them appear in the chat. So when you first show up, if there's other people there, it might take me a few minutes to get to you. So just be patient. When I call your name, you're going to unmute, hold your picture ID up to the camera. I'm going to verify it's really you. I'm going to have a quick chat with you and send you on your way. What you're going to do next is exit Zoom, open Canvas, and close everything else. All browser windows, email, everything else is closed, just Canvas is open. Take the test. My expectation is that you're going to begin the exam within a few minutes of this picture ID. So if you, let's say you do the ID verification and then you're like, oh no, I have to pee, that's okay. But don't take like a half an hour, okay? I'm going to wonder what happened to you. Start the test. Your browser activity is tracked. That means my expectation is you will not search the internet, you will not phone a friend, you will not talk to other people about the test. You're going to do it independently. And if you click out of the testing window to open a browser or to check your email or even to open a Word document, it's going to send an alert to me. So don't do that. You are allowed a calculator. You are allowed hard copy notes. You are allowed your textbook. You are allowed to talk to your toddler if they burst into the room needing your help with something, but don't phone other students or be video chatting with them. Okay. And again, don't click out of the Canvas testing window, not even to check an email. If you have a conflict with your testing time, email me. Okay. And do email me because I can work with your schedule if necessary. And I did say the wrong time, you guys. Oh my gosh, I'm so sorry. Let me just fix that really quick before it like gets burned into anyone else's brain or causes someone to have like an early heart attack. I'm so sorry. I forgot to fix that. That time is not true. Oh my goodness. It is going to be 5:30 p.m. when I open this. I apologize. And I know some of you are breathing a sigh of relief, wondering if I completely lost my mind. I have. Okay, so it is going to be tonight, in the night, much better. I'm so sorry, really am. It's been a week. Okay, so with that said, if this time is not working for you, call me or not call me, email me or talk to me in lab or just, get hold of me and we'll talk about what we can do about that, okay? Whew. I know now everyone's heart rate is up and we're all awake. Isn't that exciting? Okay, I want to talk to you about, yes, Alexi, go ahead.
It's not as important, but it also says October 26th.
Is it?
Yeah.
Lord have mercy on my soul. Oh my goodness. No one in the morning noticed that, by the way. And so they're all like, yeah, I'll take the test next October. No problem. I'm not going to see anyone in the morning class next week. They're all going to be like, next fall is when we're taking the exam. Okay. Thank you for mentioning that. I appreciate you. All right. Now, many of you already done this. Your unknown ID part 1 is finished and you've submitted your check and I'm grading them as they come in. But if you haven't, just make sure to do it by this Sunday. Sooner is better. Once you have that, you can move on to part 2. Just a reminder of what's expected for this, your notebook, all the dated entries, the image that the technician sent you in your own words, what you see, what color do you see, what shape do you see? What does that mean? What's the gram status? Okay, your analysis and the dichotomous key that you chose. What is part 2 all about? Yeah, they must have been asleep, Sean. I don't know what they were thinking. You guys are sharp. You're sharp. Okay. There's a video, watch it. The dichotomous key you're going to follow for part 2 is a decision tree. Go one test at a time. For each decision point, it's going to direct you down one path or the other. So once you're down that one path, you don't have to go back to the other side. When you request a test, make sure it's one we actually have and use the correct name for the test. There's a document called the micro test reference. That's what you will check to see if the test is available and what to call it. When you request it from the technician, make sure to put your name and ID number into the subject line. Use your school ID, school email, because if you use your personal email, sometimes it glitches and it gets blocked by the college. I don't know why, it's random, I can't understand it, but that happens. And so also, if you don't get an answer within 24 hours, resend it. Ask me for help anytime you need it. And a quick note, this is going to be throughout the process of your unknown, you're going to be requesting tests that often we have never talked about. But many of them will also, so it's kind of a 50-50 mix. But in particular, right now, we haven't really gone through metabolism. We haven't gone through metabolic testing. So there's going to be some things you're going to be asking for. You're not quite sure what they are. So I want to say one thing about oxygen preference. So some of you are going to have to determine as your first step or your second step, whether your organism is an obligate aerobe, requires oxygen to live, or a facultative anaerobe, is able to grow either with or without it. You can't just ask for oxygen preference. That's an interpretation. You need a test. The name of a test that would give you this information is called the thioglycolate test. Okay, so you can look for that. Again, there's a little bit a thing I want to do right at the end, so don't leave yet, but I just want to make sure to remind you what's due Sunday. There's a review quiz on cells. There's the last deadline for the part 1 of unknown ID, the check 1. Your lab 3 stuff is due. It's recommended you start part 2 as soon as you can. And Monday is the pre-lab 4. So do take the next few days to look over the lab 4 stuff. You also want to prep for Monday. Really important to prep for Monday. It's a fun section if you've prepped. That class will be fun. If you haven't, it's kind of a slog. So read the chapter, take the growth pre-quiz, watch the video lectures. And this is kind of a fun online textbook that I quite like. And so I would recommend looking through that as well. Make sure to make time for study for test 1. It's going to be in Zoom next Wednesday, not in October. Okay, we're going to do the cells Kahoot next Monday as kind of a, at the end of the growth section, kind of a, hey, let's review the cell stuff. It'd be kind of helpful to prepare you for the test as well. I do want to really quickly sort of touch base on these big questions for this unit. Why do microbes matter? How do we study them? What are bacteria made of? And how do they differ from eukaryotes? Okay. We did this as a speed cold call on day 1, where you were just like rapid fire, what's one word that comes to you when you hear these questions? Because we're short on time, I'm not going to do this part exactly how I would normally do it, but instead I would just want to ask you this. Would your answer to any of these questions be different today than it was that first day? Yes or no? Okay, so think about these questions for just a second and try to identify one thing that is a now more sophisticated answer that you would provide to one of these questions than what you would have been able to give on day 1. Just think about it, okay? Take a second to think about what is one thing that you could say, well, you know, my more sophisticated understanding of this question is blah. Okay. So I'm just going to quickly go through these. And if you had, if chosen that one, so if what you were thinking about was this first question, why do microbes matter? Write into the chat anything that you would say now that you would not have said on day 1. What about the second question, how we study them? Okay, Fatu says because they help us make our food. That's something she would not have said on day 1. Casey says they help in lots of processes, human and plant life, ingestion, digestion, etc. They cycle nutrients, as Kim. They can be studied with special stains and through a lot of different kinds of microscopes for question 2, says Lauren. They're too small to be seen by the naked eye, says Stephanie. They help us to treat disease. What about question 3? What are bacteria made of? What would you say now that you wouldn't have said on that first day? Peptidoglycan in the cell wall. Yeah, of course. Is there anything else you've learned that bacteria have that you didn't know before? They are prokaryotes that can survive in harsh conditions, says Danielle. They don't have a nucleus, says Kim. They're different shapes, says Fatu. Yeah, something she learned. And then what's one thing that you would have said about how bacteria differ from eukaryotes that you wouldn't have said on day one? There are a lot of similarities, but differences such as eukaryotes having a nucleus and a cytoskeleton. Bacteria differ in many ways from eukaryotes, such as the ribosome. Our human body is full of them. Yeah. Good. Okay. Yes. And eukaryotes. Okay, good. All right, everyone. So I just want to ask you now.